Hello everyone, welcome to our second lecture on cell division in the cell cycle. So today we're going to be looking at eukaryotic cell division, so focusing on meiosis, as well as eukaryotic cell cycle and regulation, as well as cancer. So meiotic cell division. So this involves sex cells, or the gametes, which are produced by meiotic cell division. It includes 2 rounds of nuclear division, meiosis 1 and meiosis 2. This results in 4 daughter cells, and each daughter cell contains half the number of chromosomes as the parent. So remember, a parent has 46, and these will have 23. Each daughter cell is genetically unique. So there's evolving a recombination of these genetics into the 23 from both parents. So meiotic cell division, so there's DNA replication takes place before prophase 1, same as mitosis. So in meiosis, in prophase 1, where the replicated chromosomes condense, and this results in 2 sister chromatids, as we can see here. 2, we have synapsis, which is when the homologous chromosomes pair with each other side by side, gene for gene, which is unique to meiosis. Here we have that pairing. The result is a 4-stranded structure called a bivalent. So here we can see our bivalent structure, where side by side. Then 3, we have the chiasmata, which is when within bivalents are visible crossovers through non-sister chromatids. So here we have our non-sister chromatids, where we can see the chiasma of the crossover between the 2 sister chromatids. So again, chromosomes first become visible as thin threads. DNA replication is already complete, like mitosis. Second, homologous chromosomes continue to condense and undergo synapsis, which is gene for gene pairing, as we can see here. The genes are paired up. And 3, when synapsis is complete, each pair of the homologous chromosomes forms a bivalent. Each chromosome consists of 2 sister chromatids. And we can see here there's 2 pairs. And again, it's bivalent. 4, the chromosomes continue to shorten and thicken, and the chiasmata between the non-sister chromatids becomes apparent. Then 5, the nuclear envelope begins to break down. So the chiasmata in detail. So key points when talking about the chiasmata is that this crossover between it occurs when there is crossover between non-sister chromatids. DNA exchange it involves the DNA exchange between maternal and paternal chromosomes. So here we have our paternal and maternal. Here we can see we have our chiasma performing, where we have crossover, and then we can see we have this exchange. So we can see we now have these recumbent chromatids that are now essentially had the randomly exchanged. Again, this is random. No nucleotides are gained or lost, so nothing is lost, it's just switching. And this results in new genetic combination, and this essentially makes every daughter cell unique. So, meiosis 1 continued. So now we're at metaphase 1. So metaphase 1, the bivalents move to the metaphase plate. The maternal and paternal homologs are randomly distributed. Then, in anaphase 1, our homologous chromosomes separate and our centromeres do not split. We see here. Then we have reductional division. Following that, we have our telophase 1, where we have 2 separate cells produced and are genetically different. So again, this is the meiosis 1. So remember, we still have meiosis 2. So again, prophase 1, which is in a later stage, the chiasmata is present. So here you have our crossing over. Metaphase 1, homologous pairs line up at the center of the cell, with bivalents oriented randomly with respect to each other. Then anaphase 1, homologous chromosomes separate, but sister chromatids do not separate. In telophase 1, the daughter cells are ready to move into prophase 2, which is the start of meiosis 2. So, note, there is no DNA synthesis between meiosis 1 and meiosis 2. So there's no new DNA made. This is still coming from those cells we had earlier. So, meiosis 2, that we talked about, resembles mitosis. So essentially, it's the exact same process as mitosis, but now it's starting from this. It's starting from the cells from meiosis 1. So the result of this, meiosis 2, is 4 haploid cells, genetically different than the parent cell. So again, haploid is 23 instead of 46. And here are 1, 2, 3, 4. So in prophase 2, the nuclear envelope breaks down and the chromosomes condense. In metaphase 2, the chromosomes align in the center of the cell. Anaphase 2, the sister chromatids separate. We can see the separation. In 4, telophase 2, the nuclear envelope reforms. So then we have our two separate cells. So comparing mitosis and meiosis. So here we have our mitosis and our meiosis 1 and 2. So we can see here that meiosis 2 essentially resembles mitosis in each phase. But again, remember, so these are identical, diploid, these are haploid. And again, we have the recombination taking place, where here there's no more further recombination. They're just split into haploid. So in meiotic cell division, division of the cytoplasm differs between males and females. So in males, we have our sperm cells that are formed. So we have our 4 haploids that become sperm cells. Each of them becomes sperm cells, where in females, only one of them, becomes the oocyte, and the others become the polar bodies, which helps support the oocyte develop. So again, we have our first stage of DNA replication. We both have that first meiotic division. Then the second meiotic division differs as one becomes our polar bodies, and one becomes the oocyte and another polar body. So meiosis is the basis of sexual reproduction. This is essentially how all reproduction takes place, at least in humans. So sexual reproduction involves meiotic cell division, which occurs in the ovaries and testes. So those are our reproductive organs. This produces our haploid gametes. So these are our sperms and eggs. Each gamete contains 23 chromosomes in humans, i.e. 22 autosomes, and either an X or Y chromosome. So that takes our female and male. But not always, but we'll see. So fertilization unites the haploid gametes to produce diploid zygote. So again, we go from haploid to diploid when the gametes unite. So remember that we have 23 from the female, so from our oocyte, and this combines with our sperm, which is also 23, forming that 46, which is completely unique as that offspring. So sexual reproduction is very important in increasing genetic diversity. It allows for quick adaptation and evolution. Regulation of the cell cycle. So cell division cannot occur at all the time. As we talked about, we have that cell cycle. If you want to go back to the lecture, there are the different phases. Uncontrolled cell division is dangerous and can lead to cancer. So essentially the basis of all cancer, in most scenarios, is uncontrolled cell division. That a cell, even though the cell itself might not be harmful, it just divides uncontrollably to where it just cannot, they can kind of be considered immortal. So cells do not proceed to cell division until they are ready. Criteria must be met. For example, DNA accurately replicated in S phase, so it's being checked for accuracy. The cells must be large enough to be able to divide into daughter cells. And if the criteria are not met, the cell halts progression through the cell cycle and usually is destroyed by apoptosis. We have regulatory mechanisms that control this. So if division occurs when the cell isn't ready, this can result in abnormal daughter cells that may not be viable and may divide inappropriately. These can see in some different conditions as well. So continuation of the regulation of the cell cycle. So progression through the cell cycle is controlled by cyclical proteins, which appear and disappear. So here's cyclin is quite a common one. So we have our CDK as well, which is cyclin-dependent kinase. Again, we evolved that kinases are involved in phosphorylation. Again, there are several enzymes that become active and inactive also in the cycles. The proteins that appear and disappear cyclically are called cyclins. The enzymes are called cyclin-dependent kinases, or CDKs. The cyclins activate the cyclin-dependent kinase, and cell division is promoted. So here, our cyclin binds to and activates cyclin-dependent kinases to control progression through the cycle. So when these are activated, we move through the cycle. This active CDK binds to its target protein, and the CDK phosphorylates the target proteins involved in promoting cell division. And we can see here the phosphorylation. Then eventually the cyclin will degrade and it will become inactive. So now a little bit more on the Cyclin-CDK complexes. So cyclin-dependent kinases, or CDKs, are always present in the cell, but are active only with the appropriate cyclin bound. So there's specific ones that are needed. So here's an example of an M cyclin, an S cyclin, and a G1/S cyclin. So there are several types of cyclins and CDKs exist at specific steps of the cycle. So as I mentioned, we have our G1/S Cyclin-CDK complex, which prepares cells for DNA replication. We have our S Cyclin-CDK complex, which helps initiate DNA synthesis. And we have our M Cyclin-CDK complex, which helps prepare the cell for mitosis. Within this, we also have checkpoints. So these are things that there's a kind of no going back, essentially, or just halt processes. So the checkpoints are to progress through the cell cycle. Certain checkpoints must be passed first. So this includes DNA replication checkpoint, the DNA damage checkpoint, and the spindle assembly checkpoint. So in the DNA replication checkpoint, which is at the end of the G2 phase, it essentially checks if all the DNA is replicated. So if something's missing or not properly replicated, is halted. In the DNA damage checkpoint, this occurs before entering the S phase. Essentially checks is the DNA damage, and again, if it is damaged, it is halted. And then in our spindle assembly, which is before anaphase, are all the chromosomes attached to the spindle. So if they're not all attached to the spindle, it will be halted. So DNA damage checkpoint. So when DNA is damaged by radiation, for example, UV rays from the sun, a specific protein kinase is activated that phosphorylates a protein called P53. So P53 is normally exported from the nucleus, therefore occurs at very low levels in the nucleus. So here, genes that inhibit the cell cycle are off. So here, normally P53 in the nucleus is not phosphorylated and is rapidly exported from the nucleus and degraded. So here we can see exported, degraded. So DNA damage occurs. DNA damage activates protein kinases that phosphorylate P53, blocking the export and degradation process. So here we have the binding that the phosphorylates P53. Now it is active, and this prevents it from leaving. Now this gene is turned on. So as P53 levels in the nucleus increase, P53 acts as a transcription factor to turn on genes that inhibit the cell cycle. So this stops division. By inhibiting the cell cycle gives the cell time to repair the damaged DNA. So essentially, we got our damage. This stops replication, so we have our stop signal. And then essentially it gives it time to repair, so certain enzymes can come in and repair. And then once this repair is done, we can move on. Genes involved in cancer. So when cell division is not properly regulated, cancer can result. So cancer is characterized by uncontrolled cell division. So how does this happen? So mechanisms that promote cell division are turned on when they shouldn't be. So essentially cells can be considered stimulated to be replicating too much, or mechanisms that downregulate replication can be affected. So it can be a mix of both or one of the others. Cells can lose the normal checks that prevent cell division, as I said. So if the cell cycle control fails, it can lead to cancer. So examples of this are Proto-Oncogenes and Oncogenes. So mutated Proto-Oncogenes may promote cancer. So Proto-Oncogenes are normal cellular genes. They're important in normal cell division. Human Proto-Oncogenes can be mutated into cancer-causing oncogenes. This can be due to environmental, chemical pollutants, and cigarette smoke. So, exposure to asbestos, molds, chemicals, various chemicals, and cigarette smoke, which is well known to cause lung cancer. So normal Proto-Oncogenes encode a variety of proteins such as cell surface receptors, G proteins, and protein kinases, and growth factors. These Proto-Oncogenes have the potential to become mutated and cause cancer. So for example, we have platelet-derived growth factor, PDGF, promotes cell division by binding to receptor kinase. This results in dimerization and results in many signaling pathways, which causes cell division to be promoted. One type of leukemia, so that's a cancer of your white blood cells, involves a mutation in the gene that encodes for PDGF receptor. The result is a receptor missing extracellular portion to bind to PDGF. This mutant receptor dimerizes without actually binding to platelet-derived growth factor and is therefore always turned on. This activates too many target proteins over a long time, and too many blood cells are produced. We also have tumor suppressors, so things that actually suppress tumor formation. So tumor suppressors encode proteins whose normal activities inhibit cell division. Tumor suppressors have different roles. They participate in cell cycle checkpoints, regress expression of genes that promote cell division, and trigger cell death, so usually by apoptosis. They act in opposition of proto-oncogenes, and whether cell division, and they also act when whether cell division occurs or not depends on both. So for cells to divide, proto-oncogenes must be on, tumor suppressor genes must be off. And again, p53 is an example of a tumor suppressor. So one example is a multiple mutation model for cancer development. So most cancers need accumulation of multiple mutations. So when several different cell cycle regulators fail, it is likely that cancer will develop. We can have gradual accumulation of mutations in multiple genes over a period of years, correlated with progression of benign to malignant forms of cancer. So for example, inactivation of first tumor by suppressor genes, for example p53, is activated, stops cell division. But eventually our proto-oncogenes become altered, and now they're oncogenes. We have the activation of oncogene, and now we can have the formation of a benign cancer that is slow growing and non-invasive. Then we can have inactivation of our other control mechanism. So now our p53 is now inactivated, and so we lose our suppressor genes. Now it becomes a malignant cancer. It grows rapidly and invades the surrounding tissues. Then we have inactivation of third suppressor, even further suppressor genes. This can result in metastatic cancer and metastasis on a new site, which is when the cancer moves to a different tissue. So that's the end of the cell cycle lectures. I hope you enjoyed it. See you in the next one.